Okay, we're going to start in about 2 minutes. Okay, it's 9 o'clock. So let's begin. So this is L7. Okay, so it's Thursday, 9 a.m., August 13th. And we're going to spend the next 2 hours talking about hemoglobin and myoglobin. Some of this material comes out of your book, Lippincott's Chapter 3. We used to do a clinical earlier in the course on sickle cell, so I used to cover material earlier in the course on sickle cell and hemoglobinopathies. And a lot of this material came out of this chapter in this book, Genetics and Medicine, which I think we're still using for the genetics portion of this book. I also, when I talk about this, refer to this this chapter in this book, Molecular and Metabolic Basis of Inherited Diseases. There are hundreds of chapters in that book. It's actually available online in the library. So if you ever want to read about some of these topics from a metabolic and molecular basis, you can do that there. Okay. So these are the objectives. We're going to go over details about myoglobin and hemoglobin, their structure, their binding sites, and how they regulate oxygen binding. Okay. What we ultimately care the most about is the conformational change that occurs between oxy and deoxyhemoglobin. And then we're going to integrate this in more detail into the physiology of O2 and CO2 transport and the role pH plays in that process. So L3 and then CC1, pH, introduction to pH, and introduction to arterial blood gases integrates with all of these topics. All right. We're going to learn something about or discuss something about the mechanism of allosteric regulation. There are theories and there are practical things that we know about this. We know that there are ligands that regulate their own binding, and then we know ligands that regulate the binding of oxygen by inhibiting the binding of oxygen and affecting their own binding. This will generally involve, excuse me, something called the Bohr effect. Here again, I mentioned this before, but we'll do it much better today. We'll talk about the oxygen cascade going from the lungs to the arteries to the veins, and the role of cooperativity in oxygen binding. Okay. We'll talk about other forms of hemoglobin, primarily fetal hemoglobin. Okay. And then we'll make this sort of very practical and applied. We'll talk about what causes shifting of the binding curve, the oxygen binding curve, to the right or to the left, shifting it so that it has higher affinity or that it has lower affinity. Okay. So a shift to the right would mean a decrease in oxygen affinity. A shift to the left will mean an increase in oxygen affinity. And then we'll talk about this in terms of specific ways of plotting dissociation curves, particularly a fractional saturation curve, something called a Hill plot. Okay. All right, so here we go. So hemoglobins are part of a, sorry, I'm going to cough again. I usually start to get congested in the middle of all of this teaching. Hemoglobins are part of a large family of heme-based proteins. Hemoglobins are globular proteins found in high concentrations in blood cells. They bind and transport oxygen from lungs to capillary beds of tissue vascular system. They're also involved in the reverse transport of CO2 and protons from the tissues to the lungs. So we're interested in the molecular structure and the regulation of myoglobin and hemoglobin. Myoglobin is a storage form that is found in the tissues where we deliver the oxygen. Yeah, the pen likes to stop working sometimes. And then we're going to integrate this into the physiology of O2 and CO2 transport and how this is all regulated by pH. So a brief introduction to the genetics of this. And as I say, this will be discussed in more detail later in a clinical correlation on sickle cell anemia. These are small groups. And in this environment today, these are in November, usually late October or early November. I'm not sure how we're going to do this. We'll do it online, presumably. We'll see how that goes in a big blue button format where you're supposed to do most of the talking. Okay, so the alpha genes, hemoglobin is comprised of, goodness, 2 alpha chains and 2 beta chains. The alpha genes are found on chromosome 16. There's a fetal form of alpha, and then there are 2 alpha genes on chromosome 16. Okay, we're diploid, so that means there are 2 copies of alpha 2 and 2 copies of alpha 1. Okay. The beta chains reside on chromosome 11. There's a fetal form or an embryonic form. Excuse me, these are embryonic forms. It's an epsilon. It combines to make an alpha beta form in the embryonic form. There are 2 fetal forms of beta chains. They're both gamma chains. They make a chain with the adult form of alpha and the different gamma forms of the fetal form. And then there are 2 adult forms of beta chain. The main form is hemoglobin A, and the minor form is hemoglobin A2, okay, delta. Okay, so hemoglobin A makes an alpha 2 beta 2 tetramer, and delta makes an alpha 2 delta 2 tetramer. All right, again, we're diploid, so all of these genes have 2 copies. Okay, so this is showing the expression of early chains on the alpha chain and early versions of the beta chain. They're gone by approximately 6 months after conception. And then the fetal form comes up. The fetal form is the adult form in alpha. So after about 6 months, that looks actually, excuse me, like 3 months. I'm misreading that. After 3 months, we're expressing alpha. The fetal form of the beta chain comes up after about 3 months. And then after birth, it goes away. So by 3 to 6 months, there's usually very small amounts, less than 2% of the fetal form. The adult form begins expression before birth, and then becomes a predominant form better than 90% after birth. The delta form represents a really small amount, 2 to 5%. And so you can know these by their larger name: hemoglobin A, adult; hemoglobin F, fetal; hemoglobin A2, the minor adult form; This form, hemoglobin A1C, is a glycosylated form. It's a non-enzymatic glycosylation of adult hemoglobin. It represents some fraction of the total, and the fraction is proportional to the amount of glucose that you walk around with. So if you're diabetic, you walk around with high concentrations of glucose, this value will be much larger. Typical adults walk around with about 5% or less. People who are wildly out of regulation in their glucose levels might have as much as 10 or 12 or more percent glucose. This lasts a long time because the life cycle of hemoglobin is typically 120 days. So it's a long-term measure of glucose regulation. Okay, and so that's shown here on this slide that's showing you hemoglobin A, the tetramer. It's showing you the N-termini, okay? And the N-termini can be glycosylated. It's an non-enzymatic glycation. I just used the word glycosylated. That's the wrong word. The correct word is glycation because it's non-enzymatic. Enzymatic would be glycosylation, okay? So they're glycated on the N-termini, okay? The predominant form of this is glucose, but you can have other forms involving fructose and other sugars. This is the adult form, but the minor form gets labeled, the fetal form gets labeled. They're just minor forms, and so you will predominantly see glycated adult hemoglobin, hemoglobin A1C. And so as it says here, they're long-term markers for diabetic sugar control. And this is discussed in the one of the metabolism chapters on diabetes. Okay. We mentioned yesterday mechanisms for looking at protein and protein composition. Okay, and I specifically mentioned SDS, polyacrylamide gels. But there are, in those cases, in that case, the migration is due to the SDS binding. Sodium didecyl sulfate is negatively charged, and it determines the mobility. You can run molecules out under non-reducing conditions. And in those cases, the protein will migrate based upon its net charge. So the total charge on the molecule, a la the counting of the protonated side chains of the various forms, will determine its mobility, okay? So if it's overall negative charged, it will migrate towards the anode, okay? And that's what is being shown here. Hemoglobin A is loaded into this little well, and then it migrates towards the anode. It has an overall negative charge, okay? If we mutate hemoglobin at some site, for example, if we mutate the hemoglobin 6 position, which is glutamic acid, to a valine, if we knock out a charge, so it's one less negative charge, then hemoglobin S will migrate slower towards the anode. Okay, so it's one less charge on the beta chain. There are 2 beta chains in hemoglobin, so this mutation, if it's homozygous, would make it a -2 charge. If this was just in one position, on average, it would be less than one-legged less negative charge. Okay. There's also a mutation known as hemoglobin C that changes it from a glutamic acid to a lysine. This is a change of -1 to +1. Okay, and so this is 2 less negative charges plus 2 more positive charge. So hemoglobin C moves even slower than hemoglobin S, and you can resolve the typical gene products in somebody by simply running them out in some kind of non-denaturing gel and look at the intensity of the hemoglobin A, hemoglobin S, or hemoglobin C band, okay? This is a measure of the presence of a mutation. This is actually what is referred to as the first molecular disease. It was predicted in the early 50s by Linus Pauling. It is now known as an anemia a genetically transmitted homozygous recessive chronic hemolytic disease. It's caused by this single mutation in the beta chain. Okay? And if you're heterozygous or homozygous, that will affect the severity of the disease. This is something we'll be talking about much more later. And so what goes on here is that this mutation induces the ability to aggregate, because this hydrophobic patch, as it's referred to, allows the hemoglobins to aggregate in the high concentration of the red blood cell. And that affects the sickle cell structure, which affects the migration of the sickle cell through the capillaries, and it actually causes occlusion. Okay, so it occludes the microvascular in the capillary bed. Okay, and so this is a slightly different rendition of this, showing blood from patients who are adults, so there's one chain, who have a significant amount of fetal, so there's AA and AF, that are homozygous for sickle cell, so they have one band, that have a mixture of adult, fetal, sickle cell, and the S chain. Notice the migration here is in this direction. The positive pole is here, okay? And then here's AS, here's SC, and here's FS, FS. All right, and so you can actually diagnose somebody by just looking at this. This is a gel. You can also do these on various chromatography devices, and you can resolve these same mutations. Notice there are lots of other mutations out there. There are hundreds of mutations. This is a very old slide. This says there's around 700 mutations. There's probably many more than that in the 24 years since I made this slide, okay? But there are lots of mutations. Not all of them cause pathologies, but some of them do. This is the most famous pathology, the most prominent pathology. Okay. So let's get into talking about hemoglobin structure. Hemoglobin has a group, a prosthetic group known as a heme. It's a specialized prosthetic group. It can be referred to as a cofactor or a coenzyme. I'm not going to make a distinction between those. Other times, books do make a distinction between what they mean by a cofactor and a coenzyme. The heme is composed of something called protoporphyrin 9. I'll show you a picture soon. And protoporphyrin 9 also has a bound iron. Functionally, the iron is in the ferrous or +2 state to effectively bind oxygen. Okay. The heme is synthesized in the mitochondria, and then it's transported and in the cytosol, and thus it's transported to the red blood cell after its successful synthesis. Okay. Nomenclature. Hemoglobin with a intact heme is a holoprotein or a holotransporter. Apo means a hemoglobin without the heme, okay? So you're not completely functional until you have your cofactor, your prosthetic group, and the folded protein. There are other heme binding proteins, cytochromes, catalase, involved in other functions like electron transfer and catalysis. It's only hemoglobin and myoglobin that are involved in oxygen binding and transport. Okay. We'll say specifically that the specific function of hemoglobin and myoglobin is determined by the microenvironment around the heme. And so the specific function in these proteins is determined by other factors in the protein fold, okay, other side chains in the specific fold of that protein, and whether they're electron transporters or whether they're enzymes, all right? Okay, and so this boils down to the sequence determines the fold, and the fold determines the microenvironment around the prosthetic group, which determines the function, okay? So here are various renditions. This one from your book is meant to simply show that it's a cavity or a cleft that it exists in. The importance of that is it's desolvated, so there's less water there. Water is actually an inhibitor in this case, so you want it to be less aqueous. You want to have a higher dielectric constant in this non-aqueous environment. This is a structure of the protoporphyrin with iron bound in the middle of the protoporphyrin. It's chelated by 4 nitrogens. Okay, so in total, this iron can have 6 bonds or 6 ligands, 4 in the plane of the protoporphyrin, and 1 above and below we'll talk about in a second. Okay. Okay, so hemoglobin and myoglobin are the 2 most abundant heme proteins in the body. The iron is held in place by 4 pyrrole nitrogens and 2 additional bonds above and below the plane of the ring. In solution, when heme is being transported around, the ligands are usually water. Once you get into the hemoglobin myoglobin, one of these bonds is due to what's called a proximal histidine. It's at position F8. F refers to a helix. It's at position F8, the imidazole on the F helix. And then the other binding is due to oxygen. So in the oxy form, okay, the zinc will have the iron. I'm saying zinc. The iron will have histidine in the back and an oxygen in the top side, okay? The interaction of the oxygen is stabilized by another histidine called the distal histidine, which is on helix E7. So that's on the E helix. Okay, so this is a rendition of a space-filling molecule of myoglobin. This is the first protein that was structurally crystallized. You can see the heme as this red disc. You can see the helices in hemoglobin, the A helix turn, B turn, C turn, into the plane D turn, E turn, F turn, G turn, H turn. So myoglobin is 100% alpha helices with turns. All right. The F helix sits behind and the E helix on the other side of the heme. You can see the positioning of the proximal histidine making a bond to the iron. You can see a molecular oxygen in the binding site, the 2 yellow balls, okay? And then there's a distal histidine that's helping to complete the formation of this binding site, all right? So one thing that came out of this early structure that came out in the late 50s was a prediction or a paradigm that globular proteins would be 100% alpha helical. You now know that's not true, but this was the only structure in the world, so maybe it's going to be true. Okay? But the principle that came out of this, in addition, when they solved the crystal structure, the high resolution structure, because then you can see all the amino acids, was that the hydrophobic residues are on the inside and the polar charge residues are on the outside or on the surface, and they're acting with water and other moieties, other macromolecules. So this is a different rendition, okay, of the protoporphyrin 9. Notice it has a slight pucker to it. It's not completely flat. The iron sits slightly out of the plane. It makes a bond to the imidazole of this histidine on the F helix. This is known as the proximal histidine. And so this will be the trigger. This will be what actually triggers sensing by the macromolecular structure, by the tertiary and quaternary fold, that there's a ligand in the binding site. When there's an oxygen up here, there'll be a slight change in position, and this will move into the plane, and this will tug on the F helix, and the entire macromolecule will know that this has happened, okay? Now, there's a form of myoglobin and hemoglobin, okay, known as MET, all right, MET myoglobin or MET hemoglobin. This occurs when the iron is oxidized to a ferric or Fe3+. This can occur by air, hydrogen peroxide, various drugs, free radicals, or it can occur in a particular mutation of hemoglobin known as hemoglobin M, okay? So since there's a ligand in the binding site with this iron form, MET hemoglobin cannot bind oxygen, okay? So that's not good. It can't transport oxygen. The sixth coordination site contains water typically, okay? So our bodies produce an enzyme that corrects this, something called NAD cytochrome B5 reductase. It's an enzyme that's in the erythrocytes that changes the iron back to a ferrous Fe+2. Okay. Now, this enzyme comes up soon after birth. So newborns don't have a lot of this enzyme, and thus they're more susceptible to met myoglobin or met hemoglobin than an adult is. Okay. So this is something that you have to look for. Met hemoglobin also binds cyanide, and thus met hemoglobin is induced. It says up here drugs. Met hemoglobin is induced by drugs like amyl nitrate. It also inhibits electron transfer. So this is one of the mechanisms of cyanide poisoning. It inhibits electron transfer. It inhibits oxygen binding. So it's trying to kill you in a couple of ways. Okay. This is simply a question that usually Dr. Bagat asks on Monday. I've thrown it in here simply because it's relevant to MET. There's a technique known as oximetry, Cox oximetry, okay? Coximetry that actually measures carboxyhemoglobin. It measures the amount of MET hemoglobin, okay? And this can be picked up in newborns, young children who have not expressed that enzyme, okay? And obviously this child has neonatal jaundice because of their early birth, okay? And this is a technique that is used in children or adults to pick this up, okay? Notice all the other choices were smoking, okay? They also contribute to this because they produce some cyanide. Okay. So let's talk about myoglobin and then get more complicated. So myoglobin is present in heart and skeletal muscle. Okay, it's an oxygen reserve. It's a carrier, a transporter. It is thought to increase the rate of transport and preserve oxygen or store oxygen in the tissue for oxidative phosphorylation. As I already said in another slide, its crystal structure was originally solved by John Kendrew in 1958. Okay. John Kendrew and others. It's a single chain, 16,000 molecular weight. It's 80% alpha helical, and it's similar in both sequence and fold to hemoglobin. So remember our discussion the other day that we're going to repeat now on sequence determining fold. There are 8 alpha helices labeled A to H. Each is terminated with a proline and a beta bend, or what is referred to as a loop. Okay. The nonpolar residues are on the interior. The polar residues are mostly on the surface for hydrogen bonding with water. Okay. So that's a solubility issue. So this is a space-filling rendition. You can see these tubes that represent the alpha helix, A, B, C, D coming at you, E, F, G, H, N terminus, C terminus, okay? Here's the heme, and here is the F helix and the proximal, and the E helix, the distal histidine, okay? So it basically is buried in between the F and the E helix. So the heme binds in what is referred to as a crevice or a cleft between the F and the E helix that is lined with nonpolar residues. The nonpolarity means that water is not happy there. So water is being excluded. Remember, met hemoglobin involves water binding. So there are approximately 80 apolar interactions from 18 residues that form the heme pocket. and thus expulsion of solvated water and hydrophobic stabilization of binding. The exceptions are these two histidines, the proximal and the distal histidine, that bind to the iron or to the oxygen. So myoglobin creates the myoglobin structure fold creates a microenvironment for heme that favors reversible oxygen binding and only rarely oxidation to ferrous iron. Oxidation of ferrous iron to ferric iron, okay? So normally we want Fe+2, and it's preventing, this microenvironment is preventing the oxidation to Fe+3. Okay. Now the fold of myoglobin, as we already have said, looks remarkably similar to the fold of hemoglobin. So this is the myoglobin structure, and this is the structure of beta hemoglobin, the adult form hemoglobin A1. Okay. So again, you can see the same structure, A helix, B, C, D coming at you, E, F, G, big turn, H, N terminus, C terminus. You can see the protoporphyrin 9, the heme, the iron, the proximal, and the distal histidine. So I'm just noticing Ernest asked a question quite a while ago. Yes, there are 4 alpha genes because there are 2 genes on chromosome 16. That means when you mutate in a hemoglobinopathy or a thalassemia sense, you have to knock out 4 copies. You have to somehow clip or remove 4 copies. And those are actually the things that happen in alpha thalassemias. That's discussed very nicely in Lippincotts, by the way. So you should look at Lippincotts for that. Okay. Hemoglobin and myoglobin sequences are remarkably similar. They fold the same. Myoglobin is 153 amino acids with 83 invariant in 23 species. So this is sort of an old comparison. We know a lot more about this than we used to, and more recent books might reflect that, or more recent reviews might reflect that. Invariant, notice means not identical. It means of a similar type, okay? Hydrophobic, okay? Hemoglobin is comprised of an alpha chain and a beta chain. They have 141 and 145 amino acids approximately, depending on the gene product. Only 15 of the 83 myoglobin sites are identical. Okay, so I'm questioning my use here of invariant. Okay, I think the invariant here is going to eventually mean that these other sites are very similar. Okay, so maybe this means the myoglobins really are identical, invariant, and that between the myoglobin and the hemoglobin, that's where you get this similar substitution. Okay, examples of these invariant or identical amino acids are the proximal and distal histidine. Both myoglobin and hemoglobin must have this to be able to bind oxygen. If those are mutated, it doesn't bind oxygen. Okay, so most changes are conserved, nonpolar. Some of the changes reflect specific changes in function. We're primarily going to talk about fetal hemoglobin. Myoglobin's a monomer, so its surface interactions are meant to be with water. Hemoglobin is a tetramer, and so its surface actions are meant to make up a polymer, a stable dimer that forms a quaternary tetramer. Okay? Changes in the alpha, beta, delta, gamma chains reflect other features of function. And specifically, we'll talk about fetal. They have the same tertiary fold from many different sequences. So here's a slide I showed you yesterday when we were talking about protein sequences. Okay, this is comparing myoglobin with horse and human chains. Okay, And it's showing you where things are identical and where things are similar if you look at residues that are not completely the same, but almost the same. Okay. I've highlighted histidines because of the pH dependence. So when you have histidines at various positions, I'm showing you those. Those are potentially buffers. Okay. And then I'm highlighting the proximal and distal histidine. Okay. So now let's switch to hemoglobin. Hemoglobin is found exclusively in red blood cells. It functions to transport oxygens from lungs to the capillary bed of the peripheral tissues. Its concentration is approximately 150 grams per liter. This allows hemoglobin to carry 1.3 milliliters of oxygen per gram of hemoglobin. So you can do the math here, how much oxygen can be transported. Okay, that's approximately 200 milliliters. Oxygen's a gas, okay, per liter of plasma. This is 87 times more than the solubility of oxygen in plasma. Okay, so without this transport, we would not be able to really provide much oxygen to our bodies. Okay. It also transports carbon dioxide and protons and something that I won't talk a lot about, nitric oxide, while buffering the pH of the plasma. The nitric oxide is discussed here, and I think you'll talk about this mostly in physiology. It's a regulator of vasodilation. Okay, so this is a space-filling rendition of hemoglobin. Notice there's an alpha 1, beta 1, alpha 2, beta 2 structure. Okay, so there are 2 beta chains, there are 2 alpha chains, and it forms a tetramer where each of the chains has a very similar fold. And you can see that in this rendition, and then in this more detailed rendition where you can see the heme and you can see the individual residues along the chains. Okay. So hemoglobin A is the major hemoglobin in adults. It's comprised of 4 polypeptides, 2 alphas, 2 betas. So it makes an alpha 2, beta 2 structure that are held together non-covalently. Okay. Each polypeptide is highly alpha helical with a heme binding pocket like the myoglobin. O2 binding in the case of hemoglobin is regulated by allosteric effectors. Okay. It's regulated by its own binding, so it's what is referred to as a positive homotrophic allosteric effector. And it's regulated by CO2 and protons and BPG. Now, we've already talked about this in the clinical stuff, and we talked about the transporting of CO2 and protons, and now we're going to be talking more explicitly about BPG. These are negative allosteric effectors because they negatively affect oxygen binding. They convert hemoglobin to deoxyhemoglobin, okay? So positive allosteric effectors affect their own binding. Negative allosteric effectors inhibit oxygen binding in this case. This is achieved by a change in the quaternary structure of hemoglobin. There's a deoxy form that favors oxygen, that favors release of oxygen, and there's an oxy form that favors oxygen binding. We're going to talk about that very explicitly. So this is a much more detailed ribbon diagram of alpha 1, beta 1, alpha 2, beta 2 hemoglobin. You can see this in ribbon diagram, so you can see a little more explicitly the 4 hemes. There's one buried in the background. The 4 hemes, okay? And the beta chains in yellow and the alpha chains in, I guess that's a dark pink, okay, or a odd red, okay? So the tetramer is actually 2 dimers. The stability of the dimers is stronger than the stability of the tetramers. And the dimers are stabilized by hydrophobic, but also ionic and hydrogen bonding. So this hydrophobic van der Waals contacts, electrostatic ionic, and hydrogen bonding comes up again and again throughout all of biochemistry. Okay. So make sure I made the emphasis correctly there. Okay. The tetramer is stabilized by hydrophobic, but also ionic and hydrogen bonding. The dimer-dimer interface, okay, so that's I actually put the wrong emphasis there, didn't I? When I said and read that. The dimers are stabilized by hydrophobic, ionic, and hydrogen bonding. That's the way I first said it, and I think that's the way it's meant. In contrast, the dimer-dimer interface, the interface that stabilizes the tetramer, is primarily ionic and polar. It's a weaker interaction, and this allows the dimer-to-dimer interface to change orientation and structure with oxygen binding. Okay, so the dimers are more or less rigid, and they rotate relative to one another upon oxygen binding. All right. The oxy form, excuse me, I see oxygen, I say oxy. The T form, or low oxygen affinity form, or the deoxy form, okay, is favored by the T conformation. Okay, so the T conformation is the deoxy conformation. It's the low oxygen affinity form. The R form is the high oxygen affinity form. It's the oxy form. Okay, so you can think of these as a conformation, T, low affinity, or deoxy. You can think of this as an affinity, R form, high affinity, or a conformation, oxy. Okay, so what do we mean by a change in conformation? All right, so in the deoxy form, there's this slight pucker in the protoporphyrin 9 in the heme. It's bound to the proximal histidine, and the iron is slightly out of the plane. Okay, it has 5 ligands. Upon oxygen binding, the oxygen fills the 6th ligand site. The iron changes electronic configuration, and that's reflected by the size of the green ball. And then the histidine moves into the plane. It chases the iron into the plane as the oxygen binding stabilizes a new conformation. Okay? And so this is the trigger for the conformational change in the entire protein. Okay? And so this is depicted here in a slightly different way in the context of the alpha helix that it's attached to. When the iron moves, when oxygen binds and the iron moves, the histidine is attached to a helix, and the helix also moves slightly. When the helix moves to the oxy conformation, you can see the sort of red ribbon moving slightly up. It changes the conformation between a junction between one dimer and another dimer. And this allows the conformational change to be transmitted to the entire molecule. Okay? So oxygen binding triggers a change in the ligation state of the iron. Ligands are binders, so the ligation state refers to 5 or 6 ligands. The histidine moves, the helix moves, the interaction between the dimer interface moves. Okay? And so this is depicted in your book in a space-filling diagram here, showing you the deoxy and the oxy form. And you can see the oxygens are represented here on the outside. This is the T conformation. This is the R conformation. Okay? There are strong interactions between the dimer, summarized here as hydrophobic and ionic, and then there are weaker interactions shown here between the dimers, okay, in the deoxy state. Upon oxygen binding, some of these ionic and hydrogen bonding interactions are broken, and the dimers move relative to one another. So this big conformational change makes the oxygen binding tighter, makes it higher affinity. This state will predominate in the lungs. This state will predominate in the tissues, okay? Because these states are in equilibrium induced by oxygen binding. So if oxygen concentration is higher in the lungs, then this is the state that will predominate. If oxygen concentration is lower in the tissue, then this is the state that will predominate. Okay? Okay, historically, T means taut, deoxy, R means relaxed, oxy. I usually simply say T or deoxy, or I say R or oxy. Okay, this is a different rendition of what's going on. This is the T state in a ribbon diagram, higher resolution. This is the R state. I don't think you can actually see, maybe you can see the red oxygen in the binding site. I think there it is right there. Okay, so it may be evident right there. Okay, so upon oxygen binding, and notice this is going from none to all. Okay, no oxygen, 4 oxygens. Okay, there's not only a motion, but actually a rotation of one dimer relative to the other. So this helix actually rotates, and you get a different rendition. You can sort of see it in that helix. It's a different rendition. You can see this helix is suddenly more towards you. The yellow helix in the back is now visible. It wasn't really visible here. So a 15-degree rotation relative to the other dimer. Okay, so oxygen binding causes a major allosteric quaternary structural change. Okay. Now the dimers, as long as it's alpha beta, the dimers are essentially identical. It's alpha, the alpha beta structure is the same, and in a typical adult, okay, so this is a matter of percentages. So I'm sorry, I think you all see these, don't you? Yes. Taylor is asking, are the two dimers identical, or is there a structural difference? The major adult forms are alpha beta, meaning A1, okay? There is a little bit of delta, and there's a little bit of fetal. And so the amount of delta or the amount of fetal in the hemoglobin reflects their percentage. If you only have 2% fetal, some of this will be gamma hemoglobin. If you only have 2 to 5% delta, some of this will be delta. So the dimers will be alpha beta, a little bit alpha delta, a little bit alpha gamma. But 90% of them will be alpha beta. Okay, and so that will be reflected in the structure. So the myoglobin monomer alpha 1 is a gene. Well, alpha 1 in this case, I'm sorry, the way you're asking this, that isn't bad. It's alpha 1 beta 1. It refers to alpha 1 beta 1 dimer, alpha 2 beta 2 dimer. Alpha gene is simply alpha. I think the two alpha genes are identical. Okay. The myoglobin monomer has one heme, and thus it's a single binding site. Oxygen binds non-cooperatively with relatively high affinity, 1 millimeter of mercury of partial pressure. So the binding is what we call hyperbolic when plotted versus O2. The hemoglobin tetramer has 4 heme binding sites. Okay, the average binding site, the P50 or the midpoint, is around 26 millimeters of mercury. So its binding is sigmoidal when plotted versus oxygen concentration. So the cooperativity reflects changes in the dimer-dimer interface upon going from deoxy to oxy hemoglobin. Okay, so this is reflected here. This is percent saturation. So 100% for myoglobin is 1. 100% for hemoglobin is 4. This is the concentration of oxygen. Notice that the concentration of oxygen in the lungs is approximately 100 millimeters of mercury. The concentration in the tissues is approximately in the 20s and 30s. Okay, so myoglobin saturates around 1 millimeter of mercury. This is in the tissues or near the tissues in the bloodstream. Once you get into the tissues, directly into the tissue, there's far less free oxygen. So myoglobin has to adjust its affinity to be able to bind the oxygen. The hemoglobin, on the other hand, saturates in the lungs, travels to the tissue, and then begins to dissociate the oxygen so it can be delivered to the tissues. So this is a reflection of the value of having a sigmoidal binding curve. The saturation is 100% at the lungs, and then as you go to the tissues and lower concentrations of oxygen, it begins to dissociate more and more of that oxygen, okay? So if we get down into the 20s, 26 is the midpoint, you're beginning to dissociate, approaching 80% of the oxygen, okay? Now we'll finish with a couple of slides just to write this down in a quantitative way, okay? So this is an equation for ligand binding, but it's written down as a dissociation constant, okay? So myoglobin dissociates oxygen. You can write down this equilibrium constant. This is a dissociation equilibrium constant in this way. You can write this as a P50, okay? So we've replaced the concentration with PO2, all right? And so we can then rearrange this to be concentration of myoglobin is equal to concentration of oxygenated myoglobin is equal to free myoglobin times the oxygen concentration divided by P50. If we then define Y as the concentration ligated divided by the total, okay, then we can substitute that equation for PMBO2 into here from the previous slide, and then rearrange this equation, canceling out the free MBs, and derive an equation that looks like this. Y, fractional saturation, looks like this. And then we can do 1 minus Y, fractional unsaturated, looks like this, okay? And then you can make this Y over 1 minus Y function, and this is what it looks like. It ends up being the partial pressure of oxygen over the KD in millimeters of mercury, okay? And so the advantage of this is we get a plot that allows you to plot the data as on a log scale. And on a log scale, log Y over 1 minus Y is log of the oxygen concentration minus log of the P50. This allows you to get log of the P50 when it crosses the axis at the midpoint. The midpoint is when this is 50-50, ligated, unligated. Okay, so this gives you the affinity. Okay, the slope of this line will be 1 because there's a coefficient of 1 in front of the slope term, okay? For cooperative binding like hemoglobin, the slope of this line will be some cooperativity term. And that cooperativity term will be called the Hill coefficient. And so we'll see a difference in this plot between myoglobin and hemoglobin. Okay, so this is where we are, and I'm going to stop here because I'm at 50:51 in the hour. So I think you've answered the questions over here that somebody asked about the T form. Okay, so the oxy form, the R form is predominant in the lungs. The T form becomes predominant in the tissues. And then we'll talk about higher order regulation of this in the next hour, which will be at 1 o'clock. Okay, so I'm going to stop the recording. This will get posted as a L7 recording, and then we'll pick this up at 1 o'clock. Okay, so see you at 1.